We are talking about mapping the brain today, cell by cell, on the K-12 Engineering Education Podcast. Engineers help us explore the brain, explains Dr. Talia Lerner, a neuroscientist at Northwestern University. Dr. Lerner recently won a Young Investigator Award from the Brain and Behavior Foundation for her research on the basic biology of addiction, depression, and other mental health conditions. I'm Pius Wong, and I had a chance to visit Dr. Lerner at Northwestern's School of Medicine in downtown Chicago to talk about the engineering in her work and to talk about her ideas about neuroscience in the future. Talia Lerner, you are faculty at Northwestern School of Medicine, and but you're not a medical doctor, right? You're a PhD?
No, yeah, I'm a PhD.
Yeah, so how does that work? How can you work in a medical school and still do what you do?
Yeah, no, so medical schools do a lot of basic science research as well as clinical research and seeing patients, and so that's essentially my responsibility, is to do basic studies in animals that hopefully eventually inform medical interventions. But I don't touch patients because I don't have an MD, so we're just working with animal models.
So you work with other people to try and answer these questions?
Yeah, and we would collaborate, for example, with people with MDs if we needed to get human samples or wanted to try to translate something into a clinic.
Okay. How would you describe the work that you do?
Yeah, so it's interesting. I mean, I think people in general care about what, mental functions and disorders that we're trying to understand. From my point of view, I'm really interested in neural circuits, and I'm trying to understand how neural circuits are set up to perform different basic functions. Mostly the circuits that I've been investigating have to do with dopamine, which we know is important for diseases like depression and schizophrenia and autism and all of these things. And part of my question is, how can there be so many different disorders that dopamine is involved in? That has such different systems, and yet they relate back to this same chemical in our brain. Yeah. And so trying to understand how that chemical actually participates in different types of processes and different neural circuits in the brain is part of the focus of my research.
In your research, I learned way back in the day that we learned about neurotransmitters. Dopamine is one of them. There's serotonin. There's a bunch of stuff. I think a lot of people might have learned that back in the day. But you also use the word neuromodulators, and dopamine is an example of one. What's the difference? Or am I behind the times now?
No, I think it's a totally fair question. I mean, it's probably one that scientists are asking themselves, because neuromodulator is not a super well-defined technical term, but I basically think of it as, so there are some neurotransmitters in the brain that are sort of fast, either excitatory and inhibitory neurotransmitters. So for example, glutamate is the main excitatory neurotransmitter in the brain. If a cell releases glutamate onto the next cell, it's going to excite that cell and cause it to fire action potentials. Things like dopamine and serotonin are neuromodulators in the sense that they don't have that fast control over neuronal firing. What they tend to do is modulate other processes that the neuron is doing to maybe up or down regulate the overall excitability of the neuron, so the likelihood that it will fire when another input comes in, or the propensity for that neuron to have its synapses change. So dopamine is a very important regulator of synaptic plasticity. So if there's dopamine around, it might even not have an immediate effect, but change the propensity of that neuron to undergo a change when other inputs come in. And so that's why I kind of think of it as a modulator rather than a transmitter.
So it sounds like you're using that, not just you, but people use that word because they don't quite know all the different things that it does. It's definitely a lot. It's not just on-off. Cells are not just turning on and turning off.
It's less straightforward than just the on-off relationship. It's more modulating the way a particular cell actually responds to those faster neurotransmitters. And that is important because it potentially changes the function of a circuit. It's potentially a way for a neural circuit to kind of on the fly switch from performing one function to another function without vastly changing synaptic structures instantaneously.
So that makes me think of one of the questions that I had listed down on my fancy paper here, but because a lot of engineers and engineering interested people are listening, and me as an engineer, I'm always thinking, okay, we taught ourselves, we learned about circuits, and as a bioengineer, we learned about, neural circuits as well. Do you know, how would you explain what the differences or similarities are between the biological circuits that you're studying and the circuits that, people know of in their house or in their car?
Right. I mean, I think people sometimes like to compare the brain to a computer, and it's an analogy that's apt up to a point, but also not. Our brains are not digital circuits that with ones and zeros, like it's not binary. And that comes out in terms of thinking about things like the signaling cascades that are activated by neuromodulators, where a synapse isn't just on or off, right? A synapse can have a certain weight to it, and that weight can change, and that weight can change on different time scales. And so there's a whole like smear of analog stuff that a synapse can do that like a node in a digital circuit isn't really capable of doing. On the other hand, they are basically
like
connections that are computing information.
The video on your website showing all the connections in 3D of your neurons is really cool looking, and it reminds me of some of the circuits that you might see in like a mechanical diagram, but still way more complicated in the brain.
Right. And so actually it's an interesting point, because the method that we used to map those circuits is basically an on-off signal. So if a connection was detected by the spiral method, then the neuron turns on a fluorescent marker, and then we see that, and we count it as like a positively labeled cell. But we're basically just counting, is a cell labeled or is it not? And what that doesn't give us is any information about the strength of the synapse that we've detected. And so we use other techniques in the lab, like electrophysiology, to go back and actually look at the strength of the synapses in a sort of more directed way. Once we have that global picture of how the brain is wired, we can form more specific hypotheses about how we think things are working, and go in with tools that actually detect more nuance.
Okay, so that is a huge question. Like, that tackles a huge area of questions that I wanted to ask about the tools that you use, because engineers, I'm sure, are involved in making them and designing them. And I mean, I guess technically you're not an engineer, or you're in neuroscience, so I assume it involves engineering and biology.
Right. I think it's kind of inevitable for neuroscience these days to have some engineering bent to what they're doing. It's part of the thing that I think drew me to neuroscience over other areas of biology, is that it just is like by necessity so interdisciplinary. Like neuroscience is a field of science that's focused on understanding an organ, the brain, and the peripheral nervous system, rather than focus on using a particular technique. So like if you said you were a biochemist, you're basically saying I'm using a particular set of techniques to understand various aspects of biology. But if you say you're a neuroscientist, you're saying I'll use any technique just to understand the brain. Yeah. And I think that's kind of interesting. It makes it a really interdisciplinary field to do research in. Right. And in particular, like in the last 10 years or so, there's been an explosion of technologies that allow us to see things in the brain that we just never could see before, and do experiments we could never do before. So it's just like a really exciting time to be a neuroscientist, but you have to keep up with the engineering aspect of the innovation in order to do some of these really exciting experiments.
Okay.
Yeah, so I would say like I'm sort of more motivated by being like a neuroscientist, but I'm by necessity also end up being somewhat of an engineer.
I get it. So you've got this end goal. I'm going to understand the brain, so I'm going to do whatever it takes, even if I don't like the computer programming,
right,
if I don't like the psychological questionnaire study, but you're going to do everything, the social science research, the computer science stuff, the biological, whatever you have to do, you're going to do it all.
Yeah, basically. Cool. Either, in some cases with collaborators who have more expertise than us, but we're going to kind of do what we need to do to try to chase down the biological questions of interest.
So what would you say are some of the more engineering type tools that you use?
Yeah, so I came from postdoctoral training with Carl Deisseroth at Stanford, who is both a bioengineer and a psychiatrist, so an MD and a PhD. And I went to his lab in part because of the tools that he's developed and that I could learn there. So two of the main things that he helped develop are optogenetics and clarity. So optogenetics is an engineering tool for the brain where we can basically use genetic tricks to put light-sensitive proteins into neurons, and that makes those neurons able to be either turned on or turned off, or in some cases just modulated by different wavelengths of light.
Is that similar to like when they used to put similar genes in bacteria and they would glow or something?
Yeah, it's exactly the same sort of recombinant DNA technology. So the GFP, green fluorescent protein gene that won a Nobel Prize a few years ago, it's the same principle. It's the sort of base protein that we've used and engineered improvements upon is a protein from algae. So algae react to light and use it to drive currents in their cells, and basically cloned an ion channel from algae that's responsive to blue light, and it allows positively charged ions to flow into the cell, which is something that just we know in neurons that turns them on. So sodium channels in neurons, that is what makes them fire action potentials. So essentially we just took this algae protein, used genetic engineering to put it into the neurons, and now when you shine a blue light, these ion channels open, positive ions can flow into cell, and that turns the neuron on. And so what that allows us to do then is like pick out really specific types of cells that we want to turn on. Before we could do this only with electrodes, where it was all just about space. So you could put an electrode in and put an electric current in the brain, and that would turn on neurons that were like near the electrode. But the brain isn't necessarily organized spatially. There's lots of different cell types intermixed within a space. And so if you can use genetics instead to pick out only neurons within that space that have an interesting property to them, that are wired up in a certain way or that express a certain genetic marker, and then you can turn only those neurons on or off, then it just allows you to do much more precise experiments in terms of the cell type you're investigating. And also because light is fast to turn on and off, you can do very temporally precise experiments, quickly turning neurons on and off.
Oh, that's really cool. And optogenetics, when I had seen that phrase, it was not something I ever studied in school. And so that's cool. It sounds like a switch, like you would turn on or off a cell like that. And you also mentioned clarity as another tool. Is that related to that?
So clarity is actually like a totally different technique. It's something that we use on fixed tissue. So tissue that is not alive anymore, that we've preserved with a fixative. But the cool thing about clarity is that we can fix the tissue into a hydrogel matrix. So you can just think of it as like a mesh grid, basically, that you've affixed all the proteins to. And once those proteins are fixed on this grid, you can suck all the lipids out of the tissue. And the lipids are the thing that are basically diffracting all the light, making the tissue look opaque. So once you get rid of the lipids, you have basically a clear brain, or whatever organ you're looking at. And that means that you can do imaging of large volumes of tissue without light diffraction or interference. Yeah, so you don't have to cut it up. We can image through an entire mouse brain just with a long working distance objective, because the tissue is quite clear. So it makes it possible for us to really examine 3D structures in a way that was very difficult before. And that's what allows us, for example, to do this, make the whole brain pictures that are on my website, where you can see, if we're looking at, for example, all the inputs to dopamine neurons, we can just see in the whole brain, get a picture of where are all those neurons located within the brain. And we're using software actually developed by other people to make those images more quantitative now and align them to a standardized atlas and assign cells to different brain regions, and really make this into like a high throughput pipeline where we can go through a lot of different animals and try to look at how all their circuits are set up and compare that to what the animal's behavior was.
Do you think they can use those same tools on human tissue, or do they do that?
Yeah, so clarity, like I said, is for fixed tissue, so you couldn't do it as a live imaging tool in humans, but it actually has turned out to work really well on fixed tissue from brains. So there are actually pretty large brain banks with tissue preserved from people who donated their brains for research, and we can get the clarity technique to work on that tissue, so we can actually go back and look at people whose brains were diseased in various ways and try to see if we can understand something about those brains, which is cool.
I had read on your website maybe it was on your website or one of your papers, a really cool phrase that stood out for me because it reminded me of engineering principles. You had said something about wanting to study like the properties and organization of neural tissue, and it sounds a lot like how engineers always say we got to study the form and the function of the things that we make. And I'm curious what comparisons you might make between the fields of engineering and biology, or engineering and neuroscience, I guess.
Yeah, I think, engineering and science are related but different pursuits.
Yeah.
In that engineering, I kind of think of as you have a goal of like a tool or a a technique or something that you're trying to make possible in mind that you're heading towards, and you try out all different solutions to get to that sort of fixed goal. Whereas science, I think, ends up being a lot more curiosity driven, where like, I have no idea what the answer to this question is, but I'm going to just like see what it is and start to develop a hypothesis as I go. So I think those are different actually ways of using technical skills to get at knowledge. But bioengineering is super relevant for neuroscience because, like, if I say, well, I really want to know what this particular cell type in the brain does, but I have no way of doing it, then now I have an engineering question. How do I do that? How do I manipulate just this one cell type in the brain? And so, like, that's, for example, the engineering problem that optogenetics helped to solve. And so, you know, I think they're just, like, people coming at neuroscience from both of those angles, from trying to ask biological questions, which is more what my lab is doing, and then from the point of view of, well, what do people need to try to answer those biological questions? And that's kind of what the neuroengineer people are doing. And they're synergistic, basically. The more tools they come up with, the more interesting questions we can come up with to ask those tools. And the more questions will be generated that need yet new tools in order to answer them. So as amazing as optogenetics is, for example, now people are starting to say, well, but, you know, we need more spatial control. We need, you know, a finer ability to cause single spikes only in a certain order in a particular pattern. We need to be able to image the activity of neurons at the same time that we're doing those activations. Like, there are all these increased demands on this already incredible technique as people get more and more sophisticated in the types of questions they can ask.
So where do you think we'll be in like 20 years when these teachers' students are already out in the field? Like, they're probably going to be interested in biology and engineering, in even social science and medicine, stuff like that. What questions do you think we're close to answering, and what questions are going to be those big, tough ones that we still got to work on in this field?
Yeah, you know, it's hard to predict. Like, I think famously, you know, when students start medical school, they get told like 50% of what's in their textbook will be proven wrong by the time they finish medical school. So I think the important thing for all of these types of education, especially in science, is that the main thing that we're trying to train you for is to be curious and to know how to structure a question. And from there, you basically have to continually keep learning in order to keep up. I think, you know, we always have these big questions of neuroscience that who knows if we'll ever answer, like, what is consciousness and do we really have free will? I'd say we're nowhere near actually answering those questions. But I think we're getting closer and closer to being able to really understand what's going on in a number of different psychiatric disorders. These are kind of interesting disorders to me because we have medicines that work at least okay. We have antidepressants, for example. We have anti-anxiety medications. But we really have no idea how to understand what effect those are having on the brain. I would say we do not understand like how an antidepressant actually works. And so I think the exciting thing is we're making progress towards really getting at how they work, but also what is depression? Are there different types of depression? Have we been diagnosing it incorrectly? Are there subtypes we need to think about? And I think that sort of stuff is going to get more and more sophisticated to the point where now, instead of, if a depressed person right now goes to their psychiatrists and asks for medication, it's basically trial and error. They just have to go through a list of medications, maybe based on some intuition that their psychiatrist has, and they, you know, one thing doesn't work and they try another one and they adjust the dose, and it takes many weeks, months, years to try to see if we can help these people. And sometimes people are resistant to all our attempts at treatment. And I think the hope that some of the research, mine and others, that people are doing now, is that you could, in the future, go into your psychiatrist to say, I'm depressed, and they could run more diagnostic tests on you. They could do brain imaging, or they could take a blood sample, or they could do any sort of neurological testing or something to try to get at, well, what type of depression do you have? Do you have a type of depression that will be responsive to drug A or drug B? And maybe we'll have new drugs to target new types of depression as we understand what those are.
And then the chemical engineers and bioengineers can get involved in that too, I'm sure. Lots of problems.
Exactly. So once you even have an idea of like, well, I've discovered this new type of depression and it ought to be specifically responsive to this type of circuit manipulation. Well, now it's kicked back to the engineers, right? Like, how do you do that circuit intervention in a human safely and effectively over a long period of time, right? Then that becomes, again, a sort of medical engineering question of how to implement it in humans.
How far do you think we are away from that being not just a hypothesis? Like, do we know that there are different types of depression that might be characterized by a different kind of circuit that you can see? Or is it just kind of like, we're just discovering this now, we don't know?
I think we're just discovering it now. I think it's more of an intuition than anything else. But sometimes people talk about it as, right now, if you look at the diagnostic criteria for depression, it's like you might sleep more or less. You might have increased appetite or decreased, right? Like, it seems like, well, this is very confusing. Basically, we're just saying something is wrong. You're not functioning properly, but we're not being very precise in terms of what's going on. And it's, to some extent, psychiatric diagnosis right now is the equivalent of saying, well, you have a headache disorder or something, right? Like, it's naming the symptoms, but it's not necessarily precise in terms of the underlying cause of those symptoms. And so I think that's the direction that we're starting to head. And hopefully, we'll make progress on this. But we know different people are, able to benefit more from certain types of antidepressants than others. The hypothesis then would be that's because there's an underlying difference in the problem with their neural circuitry that's causing the depression in the first place. But we don't know what it is. So we're trying to figure it out.
Curious and discover, I guess.
Yeah.
Hey, Pius here. I just want to take a short break to express my deepest gratitude once again to all the donors who support this show on Patreon and who support the show in other ways too. You make this podcast possible, so thank you so much. If people wanted to learn more about your lab and the specific work you do on the tools you use or just depression research, addiction research, how can they find you?
People can look up my website, which is lernerlab.org, and learn about us a bit there. And anyone is feel free to email me questions. I can do my best to answer. Maybe I shouldn't offer that before I get inundated.
I'll cut out different things, but maybe if it's okay, I'll put the link to learn.
People
just want to read your articles or something.
Yeah, people are totally free to read my website. Something that I used to write for in my postdoc that I think is great is there's a Stanford neuroblog that I wrote some articles for, but people continue at Stanford, students and postdocs, to write for that. And I think there are there's something called Berkeley Science Review. There's a podcast put out by a UCSF student called Carry the One Radio that's really good.
Like neuroscience.
So there's a lot of, I would say, accessible forms of science communication that especially students and postdocs, I think, are trying to put out there and make some of these things more accessible, hopefully.
No, that's awesome. And you know what? I just realized I skipped a big question that I should have asked you when I had heard it. You had mentioned a virus-based technology for tracing cells. And when I totally wanted to ask you about that, and I feel like engineers are involved in that.
Yeah.
What is that? And I understand you use the rabies virus.
Yeah, no, that's a really cool technique. So rabies virus, part of why it's scary is because it's able to traffic through our neural circuits and kill neurons along the way. So normally how you get rabies, you get bit on the finger by, say, a bat with rabies, and the rabies virus actually tracks back up to your central nervous system by jumping retrogradely through synapses. So it uses your sensory neurons that are projecting down into your finger to jump back through the nervous system to get into your central nervous system. So that's bad for us, but we can use that property of rabies to actually trace neurons. So if we want to know which neurons are connected to a certain type of circuit of interest, the rabies virus is really good at jumping back through those synapses. So the engineered rabies virus that we use is changed in a couple of important ways. So it's engineered so that it can only jump back one synapse, so it doesn't keep going. And the way that we do that is it depends on a glycoprotein that it makes to jump the synapses. So we make a version of the virus that does not have that glycoprotein, and then we provide it in a separate virus. So it has a glycoprotein in the cell that it starts replicating in and can jump back one synapse from there, but then it doesn't have the glycoprotein anymore after it jumps into the next neuron, and so it can't jump any more synapses. So it's restricted to one synapse. And then the other change that we can make to it is we can actually change the surface protein that it expresses, that it uses to infect cells, and replace it with a receptor or a surface protein that can only attach to a receptor that birds have, but mammals don't. So that way, if this virus is injected into a mammal like us or a mouse, it can't infect any neurons because we don't have this receptor that birds do. But we take the bird receptor and we can put it genetically, specifically into only certain cells in the mice. And then the rabies virus can infect only those cells and trace only from those cells. So we make it very specific in terms of which cell it infects first, and how many synapses it can jump. And then it becomes a very precise tool for mapping neural circuits.
Yeah, you said that as if oh, this is just a thing that happened, but I've heard 20 different engineering/biology problems in order to make that possible.
Right, there's a lot of steps, both of understanding how the virus itself works and kind of splicing in all these genes from different organisms. And
you said we make, so that's your lab? Or?
So the kind of original, I guess, engineered rabies virus was made by Ed Calloway's lab and Ian Wickersham. And then it's been improved upon or added to in various ways. So this is another thing. It's once you have a base technique, there's lots of ways you can start adding on to it. And so one thing that I worked on in my postdoc was we made it so we wanted to trace from cells not based necessarily on a genetic identity like a molecular marker, but based on their output projection targets. So I wanted to trace dopamine neurons that only projected to a particular part of the striatum that I was interested in. And so we added in another virus that could infect the axons of neurons in the striatum, trace back to the dopamine neurons in the midbrain, and use that virus to express the things that rabies needs to express, and then inject the rabies. And now the rabies tells you cells that input to cells, the output to somewhere else. So it gives you information both about inputs and outputs of neurons, which I think is really interesting.
Yeah, no, it totally is. And I can imagine there must be some flow chart somewhere saying which connections go where, and it looks just like an electrical diagram in my head when you're talking about it. And it sounds dangerous. Is that not true? Or I know that you say you put in these markers to prevent it from jumping to the mammalian tissues. I'm just curious, what do people think about it?
Yeah, it's not crazy risky. We take safety precautions, obviously, so we handle the virus with gloves and lab coats and eyewear and things like that.
Lab,
It's a research lab, so we try not to expose ourselves to the virus, but it's relatively unlikely that it would actually cause a problem because it is engineered in these ways that make it unable, for example, to infect mammalian cells in theory. It's still, for example, recommended if you accidentally inject yourself with even this modified rabies virus that you would go to the hospital and get a rabies vaccine. But overall, the risk is not insanely high. It's biosafety level 2, which means most research labs can do it with basic training precautions. There's no huge hazmat suit required or things like that.
Yeah, no, that's really interesting because it does remind me of also the engineering labs where even a lathe is dangerous if you don't know how to use it. I would assume it's the same thing for biological tools. It's dangerous if you don't protect yourself.
Right. It's just important, basically, to know the proper ways to protect yourself, but assuming those precautions are taken.
Yeah. So all the teachers listening,
nervous.
they can teach their kids safety precautions, and that would be a good skill no matter what you do. Okay, I know I ended in a very specific question there, but thank you so much again, Dr. Talia Lerner, and I hope to learn more about what you do over time.
Thanks a lot.
Thanks again to Dr. Talia Lerner, a professor at Northwestern University in the Department of Physiology. Check out some of the cool images that she makes of neural circuits at her lab's website. Links are in the show notes. For For more information on any of the people, concepts, or terminology you heard in today's conversation with Dr. Lerner, check the show notes or just visit the podcast website, k12engineering.net. Remember to subscribe to the podcast to stay up to date on engineering education discussions. Find us on iTunes, SoundCloud, the Public Radio Exchange, or any other podcast platform. Leave me some comments and reviews too if you feel so bold. Tweet the show at K12Engineering or tweet me at Pius Wong. Get more show notes and transcripts at the regularly updated website, k12engineering.net. Our closing music is from Late for School by the artist Bleeptor, used under Creative Commons Attribution License. The K-12 Engineering Education Podcast is a production of my independent studio, Pios Labs. Support Pios Labs with regular contributions by going online to patreon.com/Pios Labs. You can also just buy me a coffee. Links on how to do that are on the website and in the show notes. Thank you all for listening, and please join us next time.